once out of 15 years. That's the amount of times amoeba has actually appeared on the Leaving Cert. Now what we're going to do in this video is I'm going to talk you through Protista, and I'm going to give you my insights, my ideas about how I feel microorganisms could be important for your Leaving Cert, where I'd actually focus my energy, but then we're actually going to go ahead and cover amoeba, and you'll actually see why. So if you look at the board behind me, maybe if you even took a screenshot of that there, that is all the information you need to know. That took me a lot of time to set up, and you can see it's quite concise. The actual biology course itself, although there's loads of different things on it, as you start to pick things here and there, pick examples, crossover ideas, you can really pare it back. Okay, so first of all, what is this? This Protista, this is one of our microorganisms. Now microorganisms are part of Unit 3. Okay, so for Unit 3, any of my students will know that is our area where we start to actually diversify a little bit and get ourselves a little bit of choice. Microorganisms themselves, you would know, are part of our study plan. They're actually part 3 of our have to know study plan. Okay, Protista is one of our 4 parts of microorganisms. Okay, so one of our 4 separate parts. So we had also bacteria, we have fungi, which we will see as well, and viruses. Okay, so that's really important that you know that. In terms of what I feel will be important for the Leaving Cert in the year 2020, well, I think that you should base all your study around bacteria. You can actually study microorganisms as if they are one chapter. Now, bacteria has definitely a lot of information in it, but if you base and compare the other ideas towards bacteria, just on one sheet of paper, you will actually get a serious amount done. So I would look at bacteria, even though it came up in 2018, that's where my main focus would be. I would look at viruses as well, and that's absolutely nothing to do with what's going on at the moment. That was a prediction for myself back at the start of the year, and I still hold by it, so I really would look at those 2. And then I would definitely not leave out yeast. And the reason I wouldn't leave out yeast is because last year, as we'll see, rhizopus was such a massive part of the Leaving Cert that they asked you sexual and asexual reproduction in a rhizopus. You also probably note that that respiration wasn't on last year's Leaving Cert, apart from the experiment where they used yeast. So I definitely have a look at this and make sure that I'm comfortable with that, and we will get comfortable with that in the next video. In terms of amoeba, as I said, only been asked once. It was worth 5%. It was a question 3. It was a short question. Okay, there is not much to it. We would have actually come across amoeba already. If you remember and you rewind back to osmosis, when we studied osmosis and diffusion under the chapter of osmosis, we actually spoke about amoeba. And the actual parts of amoeba we needed to know, most of it was actually in that chapter as well. So it's really, really nice crossover. In 2019, just to remind you then, rhizopus reproduction, yeast, and then in 2018, bacteria. So I would not actually, I wouldn't cross off amoeba. 1 out of 15, it kind of has to come up again. And a couple of the mock companies also feel, they kind of seem to go with my prediction as well, that they could intermingle this microorganism chapter with an osmosis question. So just make sure that you really study those in tandem. In terms of what we actually need to know, very, very, very little, which is fantastic. We need to know this diagram. In 2010, they gave you this diagram, and they asked you to pull apart a few different things. Okay, just let me point out a number of things for you here. First of all, it doesn't matter about the actual structure of it. I've drawn it with these 2 ideas here. These 2 pseudopods, which we'll talk about in a second, and that's just good for a label. I've drawn that they have a nucleus, which is fantastic, and we'll talk about that in a second. They have this thing called a contractile vacuole, which actually makes the amoeba unique. Their main process that we study is based around a contractile vacuole. And then also they asked you to label these food vacuoles. Okay, so back in 2010 they asked you to label those. So we know a vacuole does loads of things, stores waste, stores food. In this case we're going to talk about water, but make sure that you are able to draw that diagram and definitely have a number of labels on it. In last year's question on rhizopus, you actually needed 6 labels. You wouldn't need 6 labels on this, but I just make sure I'm familiar with all these different things here. In terms of the wording, what do we need to know about this? Well, we need to know that these amoeba are unicellular. So they're made of one cell. That's all they are. Whereas you're multicellular, you have billions of cells. They are unicellular, just one. So if something goes wrong with one cell in your body, it's fine. If something goes wrong with one cell here, they're dead. Okay, but they're unicellular. And it's definitely worth going, as you base your study around bacteria and you look at the different sections, it's definitely worth your while making sure that you're okay with which one's uni, which ones are multi, and all these different words here. Second thing I have is this pseudopodia. So these pseudopodia, they actually just act as like, they just allow the amoeba to move. They act as ideas for movement. These are these parts here. So what happens is the cytoplasm pushes into them, and this amoeba can move. Almost, if you think about like how you'd see like a worm moving along the ground, quite similar in that respect. Heterotrophic. So the next word, heterotrophic. Remember, we had two ideas all the way back in ecology, in fact all the way back in photosynthesis at the start of the year as well. We said you're either autotrophic or you're a heterotrophic. Autotrophic, we would have said you automatically can make your own food. That's not technically the idea, but that's things that can photosynthesize, things that have chlorophyll. Okay, heterotrophic, like us, means that we have to get our food from outside sources. And this amoeba is exactly like us. It has to get its food, it has to source it from outside itself. So it is heterotrophic. Okay, in certain parts of microorganisms we need to go a little bit further on that, but here all we need to know is it's heterotrophic for the moment. And it's also eukaryotic. Now you wouldn't think it is eukaryotic, because I don't know, subconsciously students seem to think, oh, if it's small it must be prokaryotic, it must be insignificant. Well, the way I remember it is, you are eukaryotic. And the reason you are eukaryotic is because you have a nucleus and you have what's known as membrane-bound organelles. So membrane-bound organelles. That is a mitochondria. In a plant, a membrane-bound organelle, we just refer to like a chloroplast. Okay, so therefore they are eukaryotic because they have a nucleus and membrane-bound organelles. They don't just have DNA and RNA going everywhere, and some structures don't actually have mitochondria. So they are eukaryotic, not prokaryotic. Okay, so now we've got that introduction idea here. Let's just look at the two main ideas we need to be aware of. One is this idea of osmoregulation. So osmo, something to do with water, regulating the water. Osmoregulation. Now what actually happens in osmoregulation is, for the most part, amoeba actually live in fresh water. Okay, so they're in fresh water here. So what do I mean by that? It just means not salt water. And then what actually happens is, due to osmosis, so this due to osmosis, water actually rushes in, because we know the definition of osmosis is water going from an area of high concentration to a region of low concentration. Make sure you're okay with that definition. There are two variations on that. But what's going to happen here is, because there's a low concentration of water inside the amoeba, well, water is going to rush in. And as water rushes in, we know that the amoeba is going to expand. Now that can be good for certain cells, but if it continuously keeps rushing in, well, this is going to expand almost like a water balloon to the edge of it actually bursting. Now that might not matter maybe say in your body or in a plant cell so much, because you've got, as I said, many of them. If your whole being is one cell, that is a massive problem. So what happens is this contractile vacuole actually absorbs the excess water. So it absorbs any excess water when there's too much in here, when it's getting too full, and it goes to the edge, out here, it goes to the edge, and it expels that water. It pretty much spits it out. It bursts it out there. And in doing that, it actually controls and saves the amoeba from bursting itself. Okay, so that's what happens in fresh water. We have this osmoregulation. Now, a question they asked in 2010, and a question they asked on one of the mocks this year, so in line with the osmosis idea here, this is worth 1.25%, not a massive deal, but if you didn't know it, you probably weren't getting those 1.25%. Okay, so what they subsequently at the end of 2010 and in this, they asked what happens in a marine environment. Now, marine environment, they mean, okay, where there's salt water. Okay, so that's what they mean there. So it's just seawater or salt water. Okay, they asked what happens there. Well, what happens there is basically the concentration outside, if we have salt out here, is the same as the concentration inside, so not that much osmosis occurs. In fact, the water actually flows in between them. Therefore, there is not a massive use for this contractile vacuole. It's a quieter time for that in terms of the amoeba. So what do we need to know with that? Osmoregulation, we need to know what happens in fresh water, okay, and we need to know, does it occur in salt water or marine water? Not so much. Okay, now definitely look out for that, and as I said, you know, they could bring that in terms of one of these osmosis questions, so definitely after you look at this video, go back and look at osmosis. It's only a couple of pages and only a couple of ideas. Second idea in terms of this is food vacuoles. So how does an amoeba get its food? We've already seen it is heterotrophic, it has to source food from the outside. So how does it go ahead and get it? Well, it actually uses these pseudopods here. Okay, so these things here. Okay, it is known as a phagocyte, or it actually takes part in something known as phagocytosis. And what that is when the actual being itself literally surrounds whatever it's eating and digests it. So it kind of, for me, it's kind of akin to, I'm not sure if you've seen the Jaws, the original movie. You know that there's like a film poster, and there's this absolutely gigantic shark and a little person on top there. It looks like the shark's just going to go up and eat it and swallow it whole. That's what phagocytosis is. These pseudopods are going to swallow whatever its prey is here, absorb it into its body, so absorb it in here, and then just digest it. Okay, so as it digests it, it distributes the little pieces of food or whatever it's eaten to different parts of the cells where they're actually needed. And anything that it doesn't absorb, anything that it can't use for something productive, it egests. Now, it's definitely massively important you know the difference between excretion and egestion. Excretion is the removal of metabolic waste. So some waste that you have made in your body. So if I'm breathing out, I made this carbon dioxide in my body as a waste product of respiration. I made that water. So that's excretion. Egestion is just like not absorbed. Anything that's not absorbed that you remove, that's egestion. So for example, anything at the end of the alimentary canal, that's egestion. That's why we don't call that excretion. Okay, so to summarize before I show you some really good questions and some ideas that I'd love you to go have a shot of. Okay, to summarize, in terms of Protista, is it important? Well, it could be. Okay, so any student worth their salt is definitely going to have a look at this, and you are not going to leave it out. Once out of 15 years doesn't mean, oh, we leave it. Once out of 15 years means it hasn't been up in a decade. Okay, so we should definitely have it on our radar. In terms of what I think is massively important in microorganisms, I'm going to say base your study around bacteria. Okay, there's a lot in that, definitely. But if you then add up all the other things you need to know, it's probably around 2 chapters worth of material. So I'd study all of microorganisms as if it's 1 chapter. Okay, base it around bacteria. I suggest to look at viruses and yeast for basically the reasons I mentioned at the start of the video. Viruses haven't been up in a while. It was quite topical on a couple of the mocks. Yeast because of the respiration idea from last year, etc. In terms of this video itself, the amoeba is the only example of Protista, the kingdom, that we have to study. Base your study around knowing the diagram and the different parts. These 4 words, which we should know for the whole of microorganisms anyway. And then the 2 main ideas, osmoregulation and its food vacuoles or phagocytosis. Okay, so that's quite important there. Now, as you know already, and I've mentioned a few times, there's only been 1 question in 2010, question 3. I'm going to do it now, but before we do it, I've actually put 2 other questions up here from the ordinary level course. In most subjects, the ordinary level course is actually not worth looking at if you're doing higher level. Biology is different. The questions are different to the higher level, but the wording and the level is the same. What actually changes is just the amount of detail you have to have in your answer. So it's not a bad idea if you kind of become too familiar with the past paper questions to look at these ordinary level ones. So what I have here is that will help you with this is the 2012 question 2 ordinary level and the 2005 ordinary level question 6. They're extra questions on the amoeba, and just give them a shot using this material, and you would need a little bit more detail than the ordinary level marking scheme. However, it's definitely worth giving that a shot. In terms of the 2010 question, can we answer it? Well, we definitely can. If you want to pause the video now and get it up on the screen, that's absolutely no problem. I'm going to read through it as if you have it in front of you. But it says here, 2010, question 3, the diagram shows the structure of amoeba. Okay, so a short question, 5%. Gave you a diagram quite like my diagram up there. Wonder where I got that from. Wonder where they got those exact diagrams from as well. Name the parts labelled A, B and C. So A was the pseudopodia coming out here. B was the contractile vacuole, so the magic piece there that helped water expel. And C was actually a food vacuoles here, so that helps store food at the end. To which kingdom does amoeba belong? Protista, fair enough. Is the cell of amoeba prokaryotic or eukaryotic? Now you can either learn off that it's eukaryotic, or you can work it out that it is eukaryotic because it has a nucleus and membrane-bound organelles. Give a reason for your answer to part C. Oops. It has a nucleus and membrane-bound organelles. Give one function of A in amoeba. So our two functions, one is movement, or you could say if you want something to do with feeding or eating or absorbing food. So that's your second idea. But if they ask for one function, you only give one, even if you know two. Give one function of B in amoeba, osmoregulation, but anything to do with water and expelling water and making sure water concentration, that's fine. And suggest. Now this is very important, not a lot of students know this. The word suggest means you may know the answer from your study, but you may just have to think a little bit. And they're quite lenient with answers here. Many students leave these blank. There's going to be a load of these on your leaving cert. The student that doesn't leave them blank is the student that's going to get a H1. Okay, suggest one reason why B is more active in freshwater amoeba than a marine amoeba. Well, sure, I actually told you that. I'm suggesting one reason. The concentrations are the same, so it doesn't need to actually osmoregulate. It doesn't need to expel excess water. Okay, so this video as an introduction was Protista. Specifically, we looked at amoeba. In the next video, what we're going to do is we're going to look at fungi and show you how it's come up and what you need to know.